Good morning, my name is Ketu Patel and today I'm doing videotaping for DNA extraction. So before that, DNA is a deoxyribonucleic acid made up of two polynucleotide chains that coil around each other to form double helix. Any human and living organism contain their genetic material in the form of DNA. DNA usually presents in the nuclei of the cell. DNA is responsible for encoding the information that organism or cell can use to grow, reproduce, and development. So let's start now. I have the piece that I have soaked in distilled water overnight. Now I'm going to transfer into the mortar pestle using the spoon, and I'm trying not to get more water. Enough. Now I'm going to, and I have in the cup I have taken half cup of water and I'm adding salt into it. The purpose of to add the salt in DNA extraction is it helps to make DNA molecule neutralize the DNA molecules. It also helps to make the DNA molecule less hydrophilic, meaning less soluble in water, and also helps to detach the protein that associated with DNA. So it will help the extraction process easier. Now I'm mixing with the rod, and it's like salt is dissolved in the water. I'm going to pour into my mortar and pestle like a half, and I'm going to grind it. I have a piece that I crush using mortar and pestle, and also I have here the clean drinking glass water, and I put the cheesecloth and secure with the rubber band like this. Now I'm going to transfer the material slowly over it and let it just I'm going to let it transfer all the materials through this. So now I let it strain my piece contained to the glass, and now we have in the glass, as I can see, that the strain fluid. So what I'm gonna do next, I'm going to gently take off this because we don't need the content that we have in cheesecloth. So gently taking off that, and it goes in a trash can. So now in this liquid, I'm going to add that liquid detergent, like, or the dish soap, about the half. The purpose of the adding a dish soap is to separate for the cell lysis, basically. So it can break the cell membrane, and because of the cell wall, DNA will come out into the fluids that we have here with the clean glass rod. So yeah, I think it's about half. So, and I'm going to mix it with this, and I'll put this in the refrigerator for 10 minutes. So our DNA, basically, so it's been 10 minutes and I have transferred the solution in the tube. It's about 1/3 full, and I'm going to add the pepin powder. It's an enzyme. The function of this basically it will help to separate or isolate the DNA into from its surrounding protein. So I'm going to add it about half and mixing with the, I think it is half now, a little bit more. There you go. And I'm going to mix it with the clean glass rod gently, and make sure do not break the DNA. Do not break the DNA. All right. Now I have taken out this 91% isopropyl alcohol from the freezer, and I'm going to add about equal amount to the test tube by holding the test tube like a 45 degrees and slowly about the equal amount. As you can see, the separation of two layers. All right, and I'm going to let it set the solution about 1 minute. All right, so after 1 minute, as I can see, there's two different layers. One in the white foam layers, which is the DNA of the piece that we just extracted. It just look like a white stringy filament. The purpose of using the chilled isopropyl alcohol is to increase the yield of DNA. As the temperature increase the percentage of precipitation, so the temperature also overall affect the solubility and DNA become more insoluble. And as we can see here, the DNA is it's look like white stringy filaments that we have extracted from the peas. Thank you very much.